This clip represents a muscle fiber at rest, with the red tubes representing one myofiber and the brown tubes representing the myofibrils within. We also see a motor end plate depicted by pink Play-Doh, with Cheerios as the acetylcholine receptors. Above the myofiber, we see the synaptic knob of the axon, represented by the yellow tube.
Super slow action shot.
The release of calcium into the axon triggers the release of acetylcholine delivered by synaptic vesicles to the acetylcholine receptors on the motor end plate.
The influx of sodium to the muscle fiber creates an action potential, which travels down to the sarcolemma to the T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calsequestrin into the sarcoplasm of the muscle cell.
Go.
Almost as soon as the acetylcholine binds to receptors, acetylcholine esterase removes it and breaks it down, which causes the sodium-potassium channels to close. Then sodium is pumped out as potassium is pumped back into the sarcoplasm.
The thin filament is made up of three molecules, namely actin, troponin, and tropomyosin, represented in pink, orange, and green, respectively.
As ATP produced by the mitochondria is added to the thick filament, the myosin head shifts from a low energy to high energy state as ATP is converted to ADP.
The calcium, which has been released from the sarcoplasmic reticulum, binds to troponin, which pulls the tropomyosin off the active site of the actin.
The high-energy myosin heads leap to bind to the exposed active sites of the thin filament, forming a cross bridge. As the ADP is expended by the thick filament, the pulling motion of the myosin heads on the thin filament causes the thin filament to shorten.
At a closer look, the striation of thick and thin filaments creates bands of symmetry. These units, known as sarcomeres, are defined as the area between two Z-discs, represented here in blue. The H-zone is the distance between the ends of thin filaments, seen here in orange, within one sarcomere, and the I-band is the distance between thick filaments, seen here in black, of two adjacent sarcomeres. The A-band is the total length of thick filament within one sarcomere, and the only portion of the sarcomere that does not shorten during muscle contraction.